My name is Heather Ross, and I am very fortunate to be the site lead for the Ted Rogers Center for Heart Research at Peter Munk Cardiac Center, UHN. We have three presenters, and I'm looking forward to hearing about the science. The first is Jun Liu, who's going to present on high-throughput gap junction function measurement in cardiac diseases.
Hi, good afternoon, everyone. Today it's my great pleasure to be here to present you my work on the high-throughput gap junction measurement and drug screening work. And so at the beginning, I want to thank the Ted Rogers Center for providing such a great opportunity to bring us together to discuss this interdisciplinary research together. And so as a mechanical engineer, so my research topic might be a little bit different from all the previous work. I hope you can enjoy it. So at the beginning, I want to remind you a very brutal reality on the pharmaceutical industry. So while a great number of researchers have done the work in developing development of new drugs, so the cost of each new drug development skyrocketed in the past decade, and reports have shown that the average research spending on per new drug reached over several billions of US dollars in 2014. And one solution to reduce the cost is we revisited the FDA-approved drug repository to repurposing the drug effects and find a good solution to target the cardiac disease. So by doing this, we can reduce the time cost and labor cost and avoid the very tedious FDA approval process. And so in our project, by using the human iPSC-derived cardiomyocytes, we are targeting a personalized medicine to find the best treatment for each involved individual patient. And so now we still have the problem: how can reduce the labor and time cost in the drug development and screen the drug effects for the large number of FDA-approved old drugs. So with our specialty in the robotics and automation, we have developed a robotic system for injecting material into the cell and study their drug effects. And through the collaboration with the Dr. Hamilton in the Sick Kids Hospital, we are targeting this ARVC disease. And so one major cause of this cardiac disease is the disorders of gap junctions. So as we know, the gap junction is a special cell-cell communication channel which allows the fluorescent allows small molecules and ions to pass through freely from one cell to adjacent cells. And to manage the cardiac arrhythmia, on the market there are over 2,000 drugs that can be potentially enhance or suppress the gap junction functions. And in order to measure the gap junction functions and study the drug effects on the alteration of the gap junction functions, we have so the standard technique to measure the gap junction function is to inject fluorescent molecules into single cells and monitor how far these molecules can transfer through gap junction into neighboring cells. And however, at present, the microinjection work is mainly done manually by skilled operators. It has a problem with the low reproducibility and has a high skill requirement. And it's very time-consuming. All the previous work only showed several tens of cells can be injected. And this low sample size is far from enough to screen a great number of drugs and find the best treatment for each individual patient. And so the major challenges for microinjection is because of the small size, the cells, the small size and various thickness. And so in the XY plane through the microscope, we can know where the cell is, but along the z-axis, we don't know how high the cell is. So that in manual operation, the operator has to be careful, in order to successfully inject materials into the cell. And the second challenge is the microinjection pipette is very small and fragile. And if you are not careful enough, it might be easy to be breaked during the setup procedure before any micromanipulation can be performed. So to solve this the challenges, we have developed the robotic system that is able to inject the materials into single cells. And the system is built around a standard inverter microscope, and 40-degree freedom robotic manipulator is used to control the micropipette for delivering material into the cell. And the XY translation stage is used to carry the cell and move them in the XY plane. So the camera is used to provide a visual feedback and to build this automated system. And the digital microinjector is used to provide positive pressure. All the equipment was controlled by the system automatically. So in order to reduce the human involvement, we have integrated several automated functions to, for example, to avoid the micropipette tip breakage. In the setup stage, the system can automatically search and detect where the pipette tip is, and place the micropipette close to the cells. And after this, all this procedure only requires a user to use a computer mouse clicking the functions, and then the system will detect the micropipette tip and place it closer to the cell. And after this procedure, the relative distance along the z-axis between the micropipette tip and the cell is still unknown. So in order to detect the relative distance between the pipette tip and the cell, we have built this contact detection methods by using the micropipette tip to touch the biological cell's top membrane, so that when the micropipette tip touches the cell's top membrane, there will be a little deformation by the micropipette tip. But with our developed computer vision algorithm, the system can detect this subtle motion, and in the next step, it will control the micropipette tip to insert the tip into the cell, and then the system will trigger the microinjector to provide positive pressure for delivering materials into the cell. And during this microinjection step, there is a very interesting phenomenon. There will be a shock wave appearing around the micropipette tip to showing a successful delivery. And here's a video showing that contact detection methods and successful injection. So, as we can see from the video, after the user selects a cell, the system will automatically lower the micropipette to touch the cell. So sometimes the cell deformation is very little and hard to observe it with our naked eyes. But the system is powerful enough to detect the cell's subtle deformation and determine the cell's top membrane height, so that in the next step, the user can select as many cells as possible for injection. So to test the system injection speed, we have used 3 different types of cell lines, which are HeLa cell, HEC293, and HL1 cells. And our results showing that the injection speed is very consistent over 20 cells per minute, which allows a user to inject over 1,000 cells in an hour. And also, we quantitatively measured how much materials can be delivered into the cell during one single microinjection procedure. So in our experiment, we did the test by injecting the liquid materials into the mineral oil. So after, for example, 20 times of repetition, it will form a little droplet. And our results showing that around 3,100 pascals, it will produce about 30 femtoliters of liquid into the cell. And we also tested the injection success rate by injecting the large rhodamine dextran dye into either cell cytoplasm or cell nucleus. So we chose this larger molecule size fluorescent dye because we want to keep the fluorescent signal only within the injected site. So it won't pass through the nuclear pore complex or pass through the gap junctions into neighboring cells. So the results showing that our success rate is also very high due to the precision motion control and the powerful computer vision algorithms. And we also, after injection, we tested the cell survival rate, and it shows over 96% for both cell cytoplasmic injection and cell nuclear injection. And to measure the gap junctions, we have injected the green HPTS dye into the single cells. And so we chose this HPTS green dye because it has a very small molecule size so that it can transfer through the gap junction. By count how many cells how many neighboring cells can take in the green fluorescent dye from the injected cells, we can quantitatively measure the gap junction functions. And through the collaborations with Dr. Hamilton and Dr. Gramolini's lab, we have we are very happy to contribute to their research in discovering the TMIM43 mutation and the discovery of TMIM96 mutation, TMIM65 alteration of the gap junction functions. And so we also tested the gap junction communications across that 3 different cell lines. It showed that the cardiomyocyte cell line HL1 cells has the highest gap junction communications. As we know, it requires a high level of gap junction communication to coordinate the cells beating motion and other types of cell communications. And we also tested a drug molecule which called 18-alpha-GA, and it showed that 100 micromolars of drug can successfully blocking almost all the gap junctions. And because of the high throughput, for the first time, we can inject over several hundreds of cells and plot this histogram to show you the statistical distribution. And so recently, we have also used the patient iPSC-derived cardiomyocytes to test the drug response, and it also shows that gap junction inhibitor can downgrading the gap junction communications, and also we tested another type of drug molecules which called KEEP. So our results showing that the 10 micromolars concentration cannot successfully enhance the gap junction, and the optimal effects reaches at about 20 micromolar after 24 hours treatment. However, we also tried to get an even higher concentration, but the higher concentration of that drug will induce more toxic effects. So this project is still going on. So we're hoping that, and we are now testing even more drug molecules. That for this specific patient-derived cardiomyocytes, we are thinking to get the best treatment from our potential drug molecules and get the personalized medicine into reality by using our robotic system. So in summary, we have built this microinjection system with the robotic automation technology, and it can inject large number of cells with high success rate and cell survival rate. And more important, so this system can be used to test the gap junction, and more importantly, it is a very powerful system to deliver liquid materials into single cells. So besides the gap junction study, I think there are a large number of applications. For example, gene transfection for some other type of cells is harder to transfect. Use the traditional way, we can simply deliver that RNA or DNA molecule into the cells. And I think through the collaborations across different areas, especially within the Ted Rogers Center for Heart Research, we are aiming to find the best solution for the patient. So at the end, I would like to thank all my lab collaborators, especially the collaborator from Sick Kids, and because of the successful collaboration, I was able to bring that powerful system out from engineering lab into the clinical research lab, and then hopefully we can benefit the patients at the end. Thank you.
Thank you very much. I think we have time for one question. I'll ask a question as a clinician. Does the actual piercing of the cell in the absence of vehicle or injecting anything else change any aspects of transcription, proteomics, et cetera, just by piercing the cell with the pipette?
From this side, I think it might cause a little damage on the cell membrane, but in our experiment, we also observed that the cell membrane can seal very quickly. Okay. So that I think it
doesn't seem to have any effect.
Yeah.
Okay.